I think it is important. I think it is important to further develop a reflection, also philosophical and ethical reflection on all things that revolve around antibiotic resistance, for instance, for clinical ethics, for public health ethics, for individual responsibility that I think is getting more and more, is increasing because we get more and more knowledgeable and aware of our own role, and so on. But I also believe...
Check out my new trampoline.
I also believe that the social sciences and humanities, including therefore philosophy and ethics, can contribute, can be instrumental to find useful, context-sensitive public health means. Of course, the purpose is always that of decreasing, or limit the spread of antibiotic resistance. My presentation will follow a bit this trajectory, starting from more abstract themes and going towards more practical issues. Now, we can talk of global health and global inequalities. In the last decades, as most of us know, as we all know, fortunately, there has been an increase in access to antibiotics, especially in countries where there were less, so especially in low and middle income countries, resulting in the challenge of widening the access to antibiotics while at the same time restricting inappropriate and excessive use. Now, constructing a sustainable yet accessible model of antibiotic distribution is a global health task. Why should we so? Well, my first reaction would be to say for basic decency, for just out of decency. But let's say that there are pragmatic and moral reasons for developing international agreements designed to control antibiotic resistance. This is often called the access and excess problem. And I talk of, as you can see on top, intragenerational justice issues, namely the idea that there are certain inequalities, fairness, justice issues that we live nowadays for all of us that are living today. And now a pragmatic reason to comes from the fact that inequalities in access to health and inequalities in health outcomes contribute to the overall population burden of infectious disease and therefore to the spread of antibiotic resistance. Now, moral reasons come from the fact, as I say, the current distribution of benefits of effective antibiotics on one hand and the burden of infectious disease on the other hand is simply unfair.
There is one there already. I should remove that one.
And it is unfair not just because it is uneven, it is unfair because we know we can do something to limit this unfairness. Yes. Besides the so-called intragenerational justice issues, there are intergenerational justice issues. In plain language, it means that we know that what we do now and what we have been doing in the past has an effect on people that will come after us or people that are very young nowadays. Now, to think in terms of intergenerational justice issue, we need to assume, and we can fairly assume, I think, that current and future generations are equally important, are equal in moral status. Now, the core of this is that the use of antibiotics, the issue at stake, it leads to, is that our current use of antibiotics, medical, veterinary, or whatever else, leads to increasing pathogen resistance, and therefore a decrease in antibiotic effectiveness for future patients. If they matter as we do, then we have in principle an obligation to preserve for as long as we can antibiotic efficacy. This has also often been labeled as the tragedy of the commons, borrowing economics concept developed at the end of the 60s. As I mentioned before, one of the main areas affected ethically by this tragedy of the commons, or anyways by the antibiotic lack or decrease of effectiveness or efficacy, is the clinical world, and therefore we look at medical ethics issues raised. Now, clinicians and others who can prescribe and administer antibiotics very often, and rightly so, I would say, feel the pressure to treat those who may be perceived as particularly vulnerable. Patients are often perceived in this way, and this causes higher prescription, or high anyway, prescription rates of often broad spectrum agents to kill potential resistant organisms. Now, this perception and feelings can be really deeply rooted and are also connected to the role of doctors and other medical staff. There is a deontological imperative, a so-called obligation to do something, that is to treat, to provide the best treatment for patients. There is another way to put it is a moral proximity. Those that are in front of us are perceived as being somehow more important, so to treat them the best you can is perceived to be more important than those that are somehow distant in time or in space.
Mirko,
I think is
your sound.
You are muted, Mirko.
Mirko, your sound was lost. Could you?
Okay, so
There you go. We're back.
Okay, I didn't do anything to mute
Nope,
myself.
you didn't.
Do I keep on? Should I restart from somewhere?
You can also turn your video on again.
Yes.
Fantastic. And then if you just repeat the...
This slide?
Yes, please.
Well, quickly, it is this idea that doctors and other health practitioners, and I say rightly so, try to treat as best as they can patients that are current, they're real patients. It is hard to ask to health practitioners that feel an obligation towards the patients to take into account public health considerations, so try to decrease the use of antibiotics when they fear for their own patients, because they are usually felt or perceived as particularly vulnerable. There is a kind of moral impetus to treat them the best one can, rightly so, right? We want our doctors to behave like this. But this somehow go against the public health needs, the needs of the community. There is this conflict of interest between an individual who, of course, want to be treated as best as possible, and to sometimes, when there is uncertainty, better to get antibiotics than not getting it. One may believe or think, for different reasons, for values, for cultural reasons. For instance, I'm Italian, I well know that. Here in Sweden, I see that it's different, but we need to take into account these social and cultural determinants, I would say. And but we try to limit the use of antibiotics, for instance, withdrawing, for instance, withholding antibiotic treatment for as much as possible, or at least this is the direction that many policies are taking worldwide, locally and internationally. And there are good reasons for that. As a philosopher, as an ethicist, I have been working a bit on, and there are, here I list other colleagues better than me, in trying to justify, finding philosophical justifications for restricting the use of antibiotics, and they reflect different school of thought, from contractualists to utilitarians, deontologists, and so on. Now, I directed my research slightly on, I cover also this, but I'm more interested in what happens in society. I think that these words from Farrar, the former director of the Work on Trust, will capture this point. We can do all the science and innovation we want, but if we can't take society with us, then we won't land the science or the challenges, and we won't access the maximum number of people. Antibiotic resistance is one of those big public global health issues where you need to have people on board, people to understand what is happening, to internalize that what they do also matter. And in my research, I've been doing a bit of theoretical thinking, but also of practical work, doing empirical research in Italy, in Greece, and Sweden. And I can say that there are huge difference with the lay population, with the lay public, on how people perceive the matter, how knowledgeable they are, and their attitudes towards potential changes. So one can talk of socially responsible use of antibiotics, but it's important also to consider the social determinants of antibiotic resistance. Even in front of situations where people are quite well aware of the problem, it is very different if you have good, if the I don't find a word, but if there are good levels of help for people who are sick at home, and then they can take 1 week off without any problems, or situation in which if you can go to work for 2 days, you are fired. And it's not about the levels of awareness or many cultural values involved. It's about what is possible to do, and we need to investigate these locally, these conditions locally. This is my last slide. It is my last work. It reflects the study that is currently under review. Second round that should be published within the year, but one never knows. And is also what put me in contact with the PAR Foundation. We have been investigating the role of prosociality, assuming that prosociality in a community can determine the effectiveness of health programs. We've been talking a lot about this in connection with vaccination programs worldwide, and there's been a re-emergence of these ideas with the COVID-19 pandemic. We have investigated the highest peak of prosociality, which is altruism. And we have found out very quickly that people who show higher altruism, which is also a character feature, are those that are keener to use in a made-up scenario, are keener to use antibiotics that contribute less or the least to antibiotic resistance. Even if this comes at a higher cost, even if this comes at a higher side effects risk. But we have done this research in Sweden, which is different from other places. We need to do context-sensitive research. And for what I learned in Sweden from this research, I would say that there are a need for talk of altruism in campaigns, in in doctor-patient communication also. There are certain structural conditions to be in place. And we identified, of course, antibiotic awareness, the basics, the sine qua non condition, trust in healthcare systems, and non-discriminatory priority setting, which I think are characteristics of the Swedish healthcare. Thank you for your attention.
Thank you. I think that is very exciting. What do you think will be the next thing? Then what are you trying to find with the research?
What I'm trying to do is to extend the borders of my research, geographically I mean, and do qualitative research that we have done in Sweden and Greece, also in other countries, and also this quantitative research about the role of prosociality. But then there are also other dimensions very important to consider, like gender issues that are very important also in antibiotic resistance, to understand it better. And try not to find a winning formula for everybody, not a one-size-fits-all solution, but to do some cross-country research to find, to identify a framework that then can be used for the development of social sciences research on antibiotic resistance.
Yeah, very exciting. Gabriel, do you have any questions?
Okay,
from
so
participants?
in terms of global north and global south, can you kind of give some specifics into what can be done on both sides, from the global south and from the global north, in terms of medical education, for emerging healthcare professionals to be able to understand these ethics?
What could be done in the global south and global north?
Yes.
To understand better the ethical dimension of this. Well, I would say that there is an equal need in the north and the global north and global south to keep an open mind and have a look at what social sciences and humanities can contribute. I have found myself sometimes, or perhaps often, when people speak frankly, defending this kind of research, because antibiotic resistance, as it is such a huge global issue, is believed to be that it has to be addressed primarily and sometimes only through international collaboration coordination. This is absolutely needed. But the point is that you need to conduct this type of research and also to include what are the values and norms that regulate people's behavior locally towards the global aim. There is, I call it, or not only I call it, the pie bias, thinking that the responsibility or the role of addressing this is just in someone's hands, and that there is not a responsibility to contribute to that also from different angles. Of course, if scientists come up with, if something exciting is in the pipeline, all ethicists that work in this field will be just super happy. Still, it doesn't solve completely the problem.
Yeah.
So yeah, my answer, an open mind, include this in education programs, global south, global north, both.
Very exciting. Thank you so much for sharing your work. I think it's going to be very exciting to follow the impact that it can have on keeping down antibiotic resistance as well.
I hope so. Thank you very much, everybody.
Thank you. We have our friend Osisiogu here again, so I'm glad to see you. I think if you start to share your screen, you can go ahead with your presentation.
Good morning. I'm Emmanuel.
You are.
I'm presenting on Colistin resistance. So Colistin is a polypeptide antibiotic belonging to the family of polymyxin, and it's a natural substance which was obtained from Bacillus polymixa variety Colistinus somewhere in the 1950s. Its therapeutic use started somewhere in 1960s, and the antimicrobial agent is active against organisms such as Escherichia coli, Salmonella, Shigella, Klebsiella oxytoca, Enterobacter coaca, etc. It, however, has some organisms that are naturally resistant towards it. For instance, all gram-positive organisms, Proteus, Morganella morgani, and some few others. Somewhere in between 1960s to 1990s referred to as the golden time for antimicrobial therapy. However, colistin along this time became toxic, producing some nephrotoxic and neurotoxic disorders. Hence, the use was withdrawn. Okay, then in the 1990s, the first patient with multi-drug resistant gram-negative organism was detected. This led to the revisiting of Colistin. Then, hence, the Colistin became the first-line antimicrobial resource for treatment of gram-negative multi-drug resistant organisms. In 2010, there was reports of acquired resistance to Colistin by Klebsiella pneumoniae. And in 2012, the WHO, that's WHO, classified Colistin as critical for human health. In 2015, we got our first report of mobile colistin resistant gene 1 being detected in humans and in animals in China. What is antimicrobial resistance? In simple definition, it's the ability of a microbe to resist a medication that was initially therapeutically effective against it. Colistin in this regard is used as a last-line medicine, as a treatment option for multi-drug resistant bacterial infection. And how do bacteria develop resistance towards colistin? Two methods: through chromosomal mutation and through plasmid acquiring resistance. The development and spread of colistin resistance in bacteria is a global threat to humans, animals, and the environment. And a significant concern is that antimicrobial resistance could lead to a post-antibiotic era where antibiotics will no longer work as they used to work. Globally, there is little surveillance on colistin resistance. In Ghana, the situation is worse, as there is little effort and paucity of data for research in this area. So why did I carry out this research? I did this to carry out colistin resistance surveillance in Ghana, specifically to determine the prevalence of colistin resistant and colistin resistant bacteria in water, and then to determine the distribution of colistin resistant genes amongst microorganisms in the water and then the habitats they find themselves. How did we do this? So simply, samples were collected, culture and identifications were carried out, phenotypic determination of colistin resistance was done, and then DNA was extracted, whereby we carried out a genotypic identification or the detection of the mobile colistin resistant genes. So in the lab, what did we do? We collected water samples, carried out our cultures on the respective media, which was conducive for the growth of these microbes from the water body. Then we used the microplates or the micro dilution technique to carry out the MIC determination for detecting colistin resistance. After which we resuspended the isolates and then extracted our DNA and processed it for whole genome sequencing, where we use the epitomic cloud software to detect the presence of antimicrobial resistant genes. So what were some of our results? And then let's discuss them together. So basically, samples were collected from about 9 communities in the Volta region and from different types of water samples: the borehole, the direct tap water, stored tap water, rainwater, stream water, and well water. For the purpose of this study, the stored water refers to water samples collected and stored for some time without being to be used in late time. While the direct tap are those you collect directly from the water source and then you use them immediately. So we look at the trend as we go further. So phenotypically, the resistance was detected in about 63 samples, and then genotypically in about 34 samples. And then this gives a corresponding reports between the two. Now let's look at the trend. You would observe that this is comparing the MIC, which is the microprobe dilution technique, with the sequencing method for the presence of colistin resistance. So over here, we would see that there's an agreement between the two methods, where the phenotypic method, which is the MIC, is confirming similar results with the genotypic method, which is the presence of the PHOP gene. Okay, so however, there is some discordance where you find about 10 MIC sensitive organisms having the PHOP present, but there are some 24 MIC also having the PHOP present. So using both methods helps us to confirm the presence or absence of colistin resistant genes in water sample. There is a different study that uses the Kebibara method, but they confirm that that method is not conclusive for detecting colistin resistance. However, this study confirms that there is a linkage between the minimum in vitro concentration technique and that of the sequencing, giving us results which are similar. We also tried to sequence the organisms present in these water samples, and we found out that Pseudomonas aeruginosa, as well as Escherichia coli, Klebsiella pneumoniae, and Proteus mirabilis were the most prevalent organisms found in the water body. And the distribution of the PHOP genes was in like manner, with Pseudomonas aeruginosa being the most contaminated, or being the most involved or implicated in the spread of colistin-resistant genes, followed by Proteus mirabilis and Klebsiella and E. coli. Then we looked at the type of water bodies that are harboring these genes the most. From this study, we observed that the direct tap water, as well as the stored tap water, were the most implicated in the spread of colistin-resistant genes, as 12 out of 19 direct tap water samples collected had the PHOP gene present, and 8 out of 17 of the stored tap water had the PHOP gene present. We also tried to compare the presence of the organisms per the locations where they are found. So the communities were divided into two settings, the urban settings and the rural settings. From the findings of this study, we observed that the rural communities were having more of the colistin-resistant genes as compared to the urban communities. However, the difference was not statistically significant, indicating that the spread of colistin might be regardless of the nature of the community. You can get it in both urban or in the rural settings. The communities were broken down into about 9, and then we tried to look at the distribution of the colistin in all the communities. We noticed that communities like the Lokwe, the Godokwe, Ahoy, and the whole technical university were having the most predominant outbreaks of colistin resistance. This is not surprising, as some of these communities are close to waste dumping sites and hospital settings. So there is a possibility of organisms moving from the hospital into the community, or from the waste site into the community. So the most frequent PHOP detection occurred in isolates from the whole technical and Godokwe, as I mentioned earlier, where the gene was identified in 83% and 75% respectively. Moderate high PHOP, moderate high PHOP rates were seen in Ziavijobe, Lokwe, and Bogami. No PHOP was detected in two isolates from Akrofo. Now, the PHOP carriage across communities again indicates geographical differences in antibiotic resistant gene prevalence. Proximity to hospitals and agricultural or industrial activity may have contributed to the selection pressures at these sites. Okay, and I did mention that whole technical university and Godokwe are good examples, as these two communities are bordering the most important hospital in the Volta region, known as the whole teaching hospital. However, the small sample sizes from each location makes it difficult to draw a definitive conclusion. So more extensive sampling is needed to truly elucidate any community level trends. What are the conclusions from these findings? So basically, there is evidence of both phenotypic and genotypic colistin resistance across many of the tested isolates, though there is discordance between MIC and PHOP detection, and this highlights the complexity of resistance mechanism. Colistin-resistant organisms were cultured from direct water sources. That is the borehole, the well water, the rainwater, the direct tap, and the stored tap. And this indicates that there is environmental public health issue, not limited to a single habitat. The spread of colistin is gradual.
So we had some connection problems before.
Can you off your video, Osisiogu? Can you off your video so that you have better connection?
I think, Osisiogu, if you turn off your video, you might get... I'll help you.
I think he's disconnected. Okay, so we have 2 questions. When he's back.
Let's see. Yeah, he had some troubles with his connection. I guess this is what happens when you work in a global environment. Osisiogu is calling in from Ghana. We sit on different locations, all of us. So let's give him a minute and see if he can connect again. Otherwise, we have to keep the suspense and send. Yes, he's back. Hi.
Sorry for that.
It is okay. We can see you now again. So please continue where you were.
Okay.
And maybe you want to turn your video off if that helps you with connectivity.
All right. Please, can you see my slides?
So we're waiting. You have started to share your screen, but you need to maybe try to open your slides again. Yeah, now we do. Yep.
So I'll take the summary again. Sorry for the network.
No
challenge.
worries.
So the most frequent PHOP detection in isolates from Ho Technical University and Godokwe, where the genes were identified in 83% and 75% of isolates respectively. There was moderately high PHOP rates in Ziavijogwe, Lokwe, Bogami, and no PHOP was detected in communities like Akrofu. The PHOP carriage across communities again indicates geographical differences in antibiotic resistance genes prevalence. The proximity to hospitals and agricultural or industrial activity may contribute to selection pressures at sites like the Ho Technical University and Godokwe. However, the small sample size from each location makes it difficult to draw definitive conclusion. Based on the data presented in this report, some tentative conclusions can be drawn about colistin resistance in bacteria isolated from water sources. 1. There is evidence of both phenotypic and genotypic colistin resistance across many of the isolates tested, though discordance between MIC and PHOP detection highlights the complexity of resistance mechanism. Colistin-resistant organisms were cultured from diverse water sources, indicating this is an environmental public health issue, not limited to a single habitat. The stored tap and the direct tap appears to be a reservoir for resistant bacteria and may enable proliferation and transmission of resistant genes like the PHOP. The rural isolates had a higher prevalence of PHOP compared to urban, suggesting agricultural antibiotic use selects for resistance. However, urban sites still harbored resistant organisms, indicating that the spread of this colistin resistance is boundless. It moves across environmental settings. So overall, this study provides valuable preliminary data on the occurrence of colistin-resistant bacteria and associated resistance genes in water sources. Several recommendations can be made from this study, but a few include evaluation of colistin usage policies in agriculture and human medicine to identify any inappropriate usage that could be contributing to resistance selection pressure. Further research to clarify the mechanism of colistin resistance beyond the MCR genes, as well as factors driving the PHOP carriage and expression. Also, we can develop mitigation strategies to reduce colistin-resistant bacterial contamination of stored tap water, perhaps through improved infrastructure or water treatments. We can carry out more public health campaigns like I'm doing right now, and then authorities can enforce regulations restricting over-the-counter antibiotic sales to help curb inappropriate self-medication. Further research can be done to clarify the drivers of geographic and spatial distribution of variability in resistance patterns. We can also collaborate globally to contain the international spread of colistin resistance. These are some of the references that were used in this study. And I'm grateful to all these people for their participation in this research. Great contribution. The fight against antimicrobial resistance starts with you. Thank you for listening.
Well, thank you. Gabriel, I think you have a few
Yes,
questions
we
as
have
well.
4 questions. Okay, 3 questions. So the first one is MIC more sensitive method to detect resistance as compared to sequencing to find the genes? That's the first question. Comparing MIC more sensitive to detecting resistance when you compare it with sequencing. Do you get the question?
Okay. Yes, very clear. Okay, so MIC is not more sensitive to sequencing. MIC detects phenotypic resistance. And it is possible for an organism to not to show phenotypic resistance, yet carries the genotypic resistant gene, which MIC may not detect. So the sequencing is a higher tool to detect colistin resistance. So an organism may have the colistin resistant gene and not show it. In that situation, the MIC will not pick it up.
Okay.
I don't know if I'm making sense.
Okay, so the next question. What's the prevalence rate of colistin usage in Ghana? And which sectors use colistin the most?
Good. The prevalence rate of colistin usage currently in Ghana is at 33%. And the sectors that use it the most is the agricultural sector. They are actually abusing it, not just using it. They are actually abusing, because most poultry farms in Ghana are making use of colistin without prescription,
Okay.
directly or indirectly.
Okay. Okay. So the next question from Dr. Adela. You find in some samples colistin-resistant bacteria by the dilution method for determining MIC, but you do not see that in some samples by the sequencing method. Okay. So the question is this: does that mean that colistin resistance in those samples is caused by a different gene mutation that was not identified yet? And could
Exactly.
it be a different mechanism of resistance that was not described yet? Did you get that?
Yes. So yeah, I get it clearly. So actually the person who asked the question has answered the question at the same time. So sometimes you may see phenotypic resistance using the MIC method. However, the target genes I was looking for did not show genotypic resistance. This means that there may be other mechanisms of resistance being perpetuated by the organism. So which is not necessarily through the PHOP. We have mobile colistin resistant genes 1 to about 10. However, in this area of my research, I did not detect any of the MCR genes. I rather detected the PHOP.
Okay.
So yes, it's possible to have other mechanism of resistance being played or shown in this environment.
Okay, thank you very much. Do you see any reason why in rural settings there is more colistin resistance compared to urban setting? That's the first part. And how is the use of colistin in animals, especially pig and poultry? That's the second question. And the third question, could that be pressing factor in rural setting? Should I go again or you got the questions?
I think I got them.
Good.
Rural setting. Okay, so the first one was asking for how come we're having more of the colistin resistance in rural settings compared to an urban setting? Well, per the findings of this research, this is not so strange. The rural communities are more engaged in agricultural activities, which includes poultry, for example. Now, in these rural settings, there is shall I say, less monitored activities of these farmers. Okay, and people do practically whatever they feel like doing in the farm to save their animals, thinking of the public health effects aftermath. So in rural communities, you'll find out that most farms are located in rural environments. For example, poultry farm. You will hardly find a poultry farm within the township. And this is because we want to ensure there is no transmission of microbes from the humans in the environment to the poultry. So most poultry farmers set up the farms in rural settings, especially in the bush, in villages. And this encourages the spread of colistin resistance in those rural communities. And yes, in terms of animal use, colistin resistance is common. And this is because even from the day-old chicks, for instance, I'm using poultry as an example because I've once been a poultry farmer. So I was once a culprit of this menace until I got to understand it better. So even the day-old chicks are introduced to colistin at a very tender age, not directly sometimes, but indirectly in the sense that these drugs are added to other combinations for the treatment of certain infections. Now, most poultry farmers are not having access to the veterinary services. So they carry out their activities without regulated advice from the trusted person, but from family and friends. So yes, the spread of colistin through animals is increasing gradually. Pig farmers have also shown some complaints in this regard as well. And could it be possible to be present in rural settings? Yes, very, very possible. For the urban setting, mostly you will find colistin resistance occurring there most likely because of the presence of a hospital facility around the area. Okay, this brings about community spread and hospital spread in, out, organism being abused, buying of antibiotics over the counter without prescription. You see, these are all contributing to the spread of colistin resistance in the community.
Okay. Oh, so I think we're on time, but we have one last question. So could you please explain what MIC sensitive and resistant means? I think that's our last question. Thanks.
Okay. So MIC simply means the mobile minimum inhibitory concentration, and then the minimum inhibitory concentration has to do with the lowest concentration of an antimicrobial agent that has the ability to inhibit the growth of a microorganism. Okay, so we are looking at the lowest concentration to inhibit it. And then for the colistin, we use what we call the microbroth technique. Okay, microbroth dilution technique. So this shows us phenotypic resistance. Okay, it shows whether the organism is sensitive to the drug or not. It does not confirm the presence or absence of the resistant genes. So the MIC is the minimum inhibitory concentration of antimicrobial agents that has the capability of inhibiting the growth of a microorganism. Thank you.
Okay, thank you very much. I know I said we are the last question, but I think we have the very last question. So what type of sequencing did you use?
So I did the whole genome sequencing.
Okay, thank you very much. That's all.
Thank you so much. I'll go back to, I want to thank both Mirko and Osisiogu. We continue to do these seminars. We're going to have the next one on October 18th. We have 3 speakers on board at that time. And during the month of November, we are planning to do 1 seminar a week, and we're working with our grantees to spread awareness and talk about initiatives during the World Antibiotic Awareness Week that is taking place in the mid-end of November. So we hope you could join us then. But with that, I want to thank you for joining us today. Please continue to spread the word, and if you're interested, go to the link that you see on the screen and you can register to our upcoming seminars. After we close the seminar today, there is a very short survey to give feedback on the content. But otherwise, I wish you a really good day. Again, thank you, Mirko. Thank you, Osisiogu. And let's continue to fight antibiotic resistance. Have a good day. Thank you so much. Thanks for listening today.